Yes.
So we
Hello.
have Nichole Broderick from Johns Hopkins, who's going to be speaking on fly microbiota effects on development. And Nichole is really one of the rising stars in this topic. So I'm really looking forward to this.
All right. Do the slides look okay?
Yes.
All right. Excellent. Hello, everyone. It is still morning on my end. I'm in the United States. I'm Baltimore, Maryland, but really grateful to be joining you today. And I'm going to try to join us. So for the rest of the week, it looks like a great series of talks, and I think a wonderful opportunity. So my work is in the area of host microbe interactions, especially in the microbiome. And as Adam already gave a nice introduction to that, it's an area of really growing appreciation and interest, and I think excitement too, both by scientists and also by the public. And ultimately, it's because of our growing appreciation for just how important the microbiome is. And essentially, this is because if we think about evolution of life on Earth and even animals, this has all been done in the background of the microbiome and microbes. And so since the earliest animals existed on this planet, they've had to learn how to negotiate these interactions. And as you can see, also harness this relationship for the benefit. And ultimately, I think the public's appreciation has been this growing understanding of the myriad ways that the microbes are impacting our health and our physiology, whether it's aspects of metabolism, like what Adam talked about, immunity, which I'll talk about a bit more today, and development and behavior, which you're also going to hear about from myself and Carlos right after me. And it's just a really exciting aspect to think about how this microbes and this community of microbes that live with us can be impacting these aspects. And as you've already heard, it's the same for the fly. And so ultimately, when we look at aspects of immunity, intestinal homeostasis, reproduction, nutrition, metabolism, and feeding behavior, we realize that microbes and the microbiome is just a component of this that's augmenting and enhancing, but sometimes hurting the host's physiology. And Drosophila has really risen in the last, I would say, 10 years or so as a great model to be able to understand these associations. And so you've already heard from others who've spoken about the great wealth of genetic and genomic tools, but I'd like to talk about it from more of an ecological aspect as well, which is that flies, Drosophila melanogaster lives in a microbe-rich environment. So we call them fruit flies or vinegar flies. These are some on a kiwi fruit, actually in my apartment in Lausanne, Switzerland during my postdoc. And you can see that the fruit's still there. There's maybe some pupae, larvae coming out to pupate. There's some adult flies up here that are interacting and mating. But the flies aren't eating the fruit. They're eating the microbes that are decomposing the fruit. And so they're really attracted initially to this decomposing, rotting fruit environment. And it's the microbes that they're consuming. And for a long time, these sort of microbe-rich environments that they were associated with were thought of first in the sense of the immune response. And this idea that having to evolve and live in this microbe-dense environment, it was a really necessary component of the host to develop this robust immune response to be able to combat potential pathogens. And what I love about the fly system is that we now have a greater appreciation for the wealth of these interactions, and this idea that in one host and in one microbiome environment context, we can study microbes that might be food, microbes that are part of the microbiome and have beneficial associations, but then also microbes that might be potential pathogens. And sometimes, depending on the different state of a fly, these microbes can be exactly the same. And so it's really the interaction that's important and not so much necessarily which microbes are present. And so this is one of the reasons I love this as a model system. And so as I alluded to on the first, another reason it's really valuable is this really nice complementarity to what we see in human systems and in flies. So if we're wanting to think about this and using it as a model for understanding aspects of human health. And some of what I'm going to focus today are specifically related to these immune responses. The other nice thing about flies is that they have a relatively low complexity of microbial community. So if you look in the laboratory and in the wild flies tend to have higher diversity of microbes. but still we're only talking about tens of taxa, not thousands that we see in human and other mammalian systems. And so the beauty of this is it means that we can have a reduced complexity of a microbiome with a host that we can manipulate both. And we don't have to artificially reduce that complexity. So we don't have to like get rid of half of them to be able to understand that, or only put back a subset, although these are things we can do with this tool. But often this can complicate when we're trying to understand what specific microbiome features might be impacting these different phenotypes that we're interested in. And as Adam had already alluded to, another great tool is that we can raise these flies azenic or germ-free without their microbiome. And essentially what we do is we take them at this embryo stage and we, as Adam said, decorionate. So it's simply putting them in household bleach, about the concentration you would get when you buy it in the market. Get rid of the outer chorion where these microbes are attached, put them into sterile environment, so autoclaved food and vials, and rear them in a sterile environment. And as long as we give them the proper levels of nutrition, you can do this for generations. And so this has given us a great and powerful tool to be able to assess the extremes. So what does a fly look like with no microbiome versus one with a microbiome? And essentially what that's shown us is that you can impact almost all aspects of physiology. So I've listed some here in some of the labs. And during my postdoc, I did a transcriptomic approach where we tried to understand what, in terms of gene expression, the gut was affected. And ultimately, we found that the microbiome was touching everything, especially developmental pathways, immune pathways, homeostatic pathways, and metabolic pathways. And so it's not surprising then that it can have this diversity of phenotypes also on the host, some of which you've already heard about. So two that I specifically looked at during my postdoc were aspects of gut morphology, where we found that there was a big impact on stem cell proliferation. So activity of stem cells in the gut, which renew the tissue, is reduced in flies that don't have a microbiome. We also found there was a big impact on gut length and cell spacing. So there wasn't a change in the total number of cells in the gut, but there was a change in the proximity of cells in the gut, so that you had them much more tightly packed in a fly that has been raised with its microbiome versus one that hasn't. And this actually has a consequence that the guts are 40% longer, which is quite impressive if you think about this in terms of what happens to the actual tissue itself. We also found that there was an impact on cell composition in the gut. So in a fly gut, you have a stem cell that asymmetrically divides to give rise to a stem cell, an enteroblast, which then differentiates into either enterocytes, so the really large main cell type of the gut, or enteroendocrine cells. And what we find is that in the absence of the microbiome, you get a shift of these enteroblasts differentiating into enteroendocrine cells. So there's a higher number of enteroendocrine cells in a fly that doesn't have a microbiome. We don't really know the physiological consequences of this yet. It's something that we're still trying to understand, but it's at least something that we can note, that there's this big impact of the microbiome on the cell architecture and aspects related to it. Another big aspect that I looked at during my postdoc was the impacts that you see on the immune and homeostatic responses. And this is especially through the main immune pathways in the gut. And so this pathway recognizes bacteria through the peptidoglycan in the cell wall and cell membranes. This interacts with the receptors, peptidoglycan recognition proteins, and then induces the NF-kappa B pathway, so canonical NF-kappa B pathway. So not only is the microbiome important for inducing the basal level of this pathway in the gut, but it's also really important for inducing what I'm showing here in red, which are the negative regulators of this pathway. And so this is essential so that a fly, just like in humans and other animals, doesn't actually have an overactive immune response to the microbes that are in the gut. So this is really important for maintaining this homeostasis. And we see in things like aging that this can start to be dysregulated. And so these pathways are really important for this interaction, not only with pathogens, but now we know and have a greater appreciation for the interaction they have with the microbiome, and in particular, the IMD pathway. And so one of the first questions that we asked is, well, what happens if these pathways aren't present? So if you don't have an immune response, what is the impact of that? And what we see is that this has given us a greater appreciation for this sort of feedback between the gut microbiota and immune response. So the microbiome is important for establishing the immune response. But in turn, the immune response is important for controlling the density. So we see more bacteria in flies that don't have a functional immune response. And it's important for impacting the composition. So if a fly doesn't have an immune response, we see an increase in diversity in the members, but more importantly, we see an increase in members that aren't normally present. And so we suspect that it allows the invasion of perhaps not as beneficial of microbes into the community. So the immune response is really important for controlling this diversity. It's also important for impacting where the microbes are in the gut. So this is an adult fly gut from the anterior to the posterior region. And what we've done here is we've stained bacteria with a dye. And you can see that in a normal fly with a normal competent immune response, we see this nice accumulation of bacteria in the posterior midgut. And we can look at this by qPCR as well, where we're just measuring abundance of microbes. In a fly that lacks a functional immune response, what we see is not only is there a lot more signal, so there's a lot more bacteria, but we lose this spatial restriction. And so bacteria are present all throughout the midgut. And we suspect that this has some impacts then on other aspects of homeostasis with the fly, because it's no longer able to control where these microbes are. And we can know that even beneficial bacteria, if they're in the wrong place, can be very problematic for a host. So this has established this nice interaction where we know that host immune responses are important for establishing these sort of basal levels and functioning of the gut, but the immune response also feeds back and is important for controlling this microbiome. As I set up my lab a few years ago, so I was first at the University of Connecticut, and just this summer, in the midst of the pandemic, I've moved my lab down to Johns Hopkins. We've been trying to explore these microbiome-mediated phenotypes in more detail. And so I'm going to go into detail a bit more about what we've been looking at in some aspects of the host immune response, but also talk about how the microbiome and microbe-microbe interactions can impact and influence behavior. So the first story I'm going to tell you about, I think, is also a nice example of how fly systems can be useful for comparative studies, including for some that are important for vector biology, for example. So this is a story of two flies. So here's Drosophila melanogaster along with tsetse fly. And so what this study came about was actually through kind of a fortuitous collaboration with a colleague of mine. And where on our end it started was we were really interested in aspects of how the microbiome might impact susceptibility to pathogens. And so there's different ways we can infect microbes. We can give them orally. And so we can just put them on the food and the fly will consume them. So we can look at sort of what happens with direct interactions with the gut. But we can also directly introduce them into the fly's body cavity, into its hemocoel. And so that's what we're showing here, where you see this septic injury model, where you can introduce microbes directly into the fly bloodstream. And so what we were really interested in was there an impact of the microbiome on susceptibility to a pathogen that we use called ECC15. So what we found is if you rear flies with a microbiome, they have a pretty low susceptibility to this pathogen. They're usually able to control the infection, they have a competent immune response, and they can clear the infection. To our surprise, though, we found that if you have flies without a microbiome, they were dramatically more susceptible to this bacteria. And so my graduate student, Rose, started to try to understand what was happening there. Because if we think about the immune response, there could be a couple of different things. So I've already told you about this humoral side, which is the production of things like antimicrobial peptides, and it's really important. But there's also a cellular component to the fly immune response. And so we initially looked at things like the IMD pathway, which I already showed you. The gut microbiome is important for inducing those levels. And so it's no surprise that we see again here that in azenic flies with this systemic model, they have a much lower immune response. So one possibility is we're putting these bacteria in, and a fly that doesn't have a microbiome with its lower immune response doesn't clear the infection. And this might be why they're succumbing to the infection. But as I said, we were really curious as to what was happening on the cellular side. And that was the question my student asked was, could the microbiome also be impacting the cellular immune response? And we looked at this first by, so the cellular immune response includes things like hemocytes that can phagocytose bacteria. They have clotting factors that are important for coagulation. But then there's this arthropod-specific melanization response. And what this involves is phenyl oxidases, which then lead to melanin production. And melanin in this system is important for both clotting wounds, but also it's a microbial so it releases reactive oxygen species and things of that nature. And so we did this by looking at measuring phenol oxidase activity as a proxy for the melanization response. And so if you take a wild type fly and introduce a bacterium to a wild type fly with a microbiome, you see this beautiful induction of a melanin response. So they're producing phenol oxidase, which we can measure on a plate reader, and we see that they have this robust melanization response. If you take mutants in this pathway, so this is a mutant called black cell, which can't do the initial prophenol oxidase conversion, you can see that it does not have a melanization response. What was quite striking, though, was if we took flies that had a microbiome and we did this same infection that you see here, they have a very low to, in some cases, it can be as low as certain mutants where they're not inducing this melanization response. And so we were really intrigued by this. And Rose and I began planning experiments for her next steps of this project. And literally at about the exact same time, I got a phone call from a colleague of mine, Brian Weiss, who works with tsetse fly. And he's a scientist at Yale in the School of Public Health. And he contacted me because they had seen an interesting phenotype with their tsetse flies. And so he wanted to explore possibilities of collaboration and comparing this and what was happening with Drosophila. So for those of you who maybe don't know much about tsetse, it's a fascinating insect. It has an important obligate relationship with a bacterium called Wigglesworthia. It has a number of different microbes that it's associating with. What's really intriguing about tsetse is that it gives live birth. And so it has a developing larvae that just comes out at the very end, pupates, and then goes on to the next generation. And so these bacteria are essential because the mother uses them to produce the nutrients that she feeds the larvae as it's developing. This is a useful tool, though, because it also means that you can feed the mother antibiotics to cure the larvae of the bacterium and then look at the impacts of the lack of a microbiome. So they can't do the way we do, where they bleach the chorion because of how the reproductive cycle is, but they can use antibiotic feeding as a way to test this. And when Brian and his team had done this, what they noted was these insects that are aposymbiotic, so don't have microbiome, are immunocompromised. And in particular, they lacked hemocytes. They did an RNA-seq experiment to try to see what sort of genes might be regulated from the transcriptome level. And one of the most highly regulated genes they identified was an odorant binding protein, which is kind of weird to think about why an odorant binding protein might be playing a role in this. So they began investigating whether this had a direct impact on this response. So in tsetse, you can do RNAi. So they were able to knock down using RNAi, the expression of this odorant binding protein, and found that these flies have reduced survival after sterile injury. So if you prick them sterilely, so you're not introducing bacteria, you can see in red that these flies succumb much faster than you do to flies that either the control or the rescue. And largely this is because what's happening is these flies are unable to properly heal the wound that occurs. And so they're basically continuing to bleed out and they die of both a dehydration response and a loss of bodily fluids. This melanization difference led them to check on whether the melanization response was altered. And you can see that these knockdown flies have a reduced level of phenol oxidase activity based on Western blotting. A trick that we've learned from Drosophila is that where this has been very well studied in terms of different cell types, is that cells that are important for this melanization response are crystal cells. And crystal cells are one of the three main cell types. And we can look at crystal cells by basically taking larvae, heating them up to a higher temperature. This causes the crystal cells to burst. And then you can basically see those melanization deposits where the cells have burst through the cuticle. And so what they saw was that these knockdown flies have a greatly reduced number of these crystal cells, which would correlate with the phenotype they see with lower phenol oxidase levels and also a reduced melanization response. This is where, though, the limitation of their system, so they have a great vector model, they're able to do a lot of really nice work, but the question was, is this something that they could look at more in terms of genetic approach? And this is where we came in, because it turns out that Drosophila has an ortholog of OBP6 called OBP28A, which we've now been able to show with our collaboration with Brian has a similar role. So if we look at OBP28 expression, we see flies that lack a microbiome have much lower expression levels of this odorant binding protein. They also have lower expression of lozenge, which is the transcription factor I had in the previous slide that's important for differentiation of prohemocytes to crystal cells. They have a lower level of there's the pathway there. They have lower levels of phenyloxidase and the lower numbers of crystal cells, which you can see through this cell burst assay. They also recapitulate the phenotype with reduced wound healing. So if we look at control versus an RNAi or a CRISPR line, we see that these flies are not able to melanize a wound as well as their control counterparts. They also are more susceptible to this sterile wounding where they then dehydrate and bleed out. And so this has really led us to continue to try to figure out what's happening with the system, because it's quite fascinating that in two insects we have this same situation where the microbiome is important for inducing this odorant binding protein, which then plays a role in hematopoiesis and melanization response. And so some of the questions we're continuing to explore is how the microbiome is doing this. Is it something that's direct or indirect? So if we look at expression of odorant binding protein, it specifically appears to be associated with the olfactory tissues. And so it's intriguing to think about these microbes in the gut, how they're influencing this response that's happening in so further away. So it's sort of more in the sensory cells. So we're trying to understand this mechanism, whether it's a direct, so is it a microbial factor that's inducing this, or is it perhaps indirect, such that the microbiome is inducing another pathway that then feeds into this odorant binding protein response. We also just have a really big question that we're trying to understand is why, in general, why evolutionary is an odorant binding protein having a role in cellular immunity. And this is especially intriguing to us because if you look at the olfactory response of these odorant binding protein mutants, they're not altered in their response to odor signals. And so odorant binding proteins, their role basically is to interact with hydrophobic molecules, bring them to olfactory receptors. And so in the case of OBP28A, which is actually the most highly expressed odorant binding protein in the fly, it's present in all the Basiconic sensilla. If you knock it out, it actually doesn't alter their response to odorants. And so it's really intriguing to try to wonder why, what the role is of this. Is this a sort of modern role or perhaps a more ancient role of this odorant binding protein? We're starting to get some clues more into what's happening. So if we look at these crystal cell defects with OBP28A, we can see that both azenic flies and OBP28A mutants lack these crystal cells. And they also, as larvae, lack the ability to have these melanotic wounds. So it's not just an adult response, but also a larval response. And you can see that quantified over here. And so this is intriguing because this is an azenic fly that has normal levels, or that it does have some level of expression of OBP28A. And then in this case, you have a mutant for OBP28A, but it has a microbiome. And so now we're trying to figure out where those are intersecting to have this impact. I would also note the OBP28A mutant is always more affected in this response than we see in the azenic fly. So it gives us a nice way to sort of amend those responses. So one of the beauties of the fly system is that the microbiome members are also basically all culturable. And so what this means is that we can take the flies, remove the microbiome, but now try to put it back so that we can see what microbes might be important for establishing this response. And then also we can look at mutants and things like that in the future. So we've gotten a little bit of a handle on what might be happening here. And so what we've done is we've done this in two different ways. And that's that we've taken the microbiome members, introduced them. So basically put them in with the embryos when we first dechorionate, and then let the adults emerge and sample them. And what we find is if you look at those initial adults, we don't see a restoration of the expression of OBP28A. If we reassociate the microbiome with adults, though, and we look at the next generation, there we see in the larvae that have emerged from these adults that we do see restoration of OBP28 expression. And so what this tells us is that there's a developmentally dependent association of this microbiome. And we think specifically what's happening is it's important at this point where you have the adults establishing the microbiome and whatever expression is necessary for the embryo, and that it's impacting this first wave of hematopoiesis that happens in the embryo. And so that's why you need to have it in the prior generation, and it can't just be in the associating with the larvae, because even though they go through another wave of hematopoiesis as larvae, this is where the important interaction appears to be. And so we're trying to understand this in more detail, and specifically what factors might be influencing this. So I'll quickly wrap up by thinking about why odorants and why other ways odors might be important. And that's by what we've looked at by looking at different microbe interactions in the system and how it could influence behavior. And so this came out of work from a graduate student, Caleb Fisher, where he was just curious about whether microbiome members could influence Drosophila attraction. So he developed, he took the classic t-test or T-arm test, where you basically put in an odorant and you give flies a choice. And if they move towards the odorant, they're attracted. If they move away, they don't like it. And so if you give them something like apple cider vinegar, which they love, they'll go towards the test arm. If you give them benzaldehyde, which they don't like, they'll avoid it and move away. And so he basically then developed an apple juice media so he could grow the microbes in sort of a more natural inoculum source, and then tested a variety of microbes from our system, both wild caught and laboratory flies. And so you can see that the lactic acid bacteria were slightly, they avoided slightly, or really had no significant effect, whereas acetic acid bacteria, and especially the yeast, were quite attractive to the fly. The next question Caleb was interested in is, do microbiome interactions impact this? So he had initially looked at all of them growing alone. And now the question he wanted to ask is, what happens when different microbes go together? Because we know on the fruit and in even the fly tube, there's interactions happening. And so he decided to start with one acetic acid bacteria and one yeast. And he tested this in two ways. One is he grew them separately and then mixed them so that they grew to produce their own metabolites and things like that, and then just did a mixture of the two, versus what happens if they grew together. So they grew together the entire time of their duration, and then you sampled that looking at the different odorants and then what their response was. And what he found is the combination of microbes growing together was always more attractive than if they were grown separately and mixed. And there was sort of a maximal attraction at these later time points, around 100 hours or so, where flies were most attractive. So if you compared 48-hour grown together mixture to a 96-hour, they were more attractive to the 96-hour, for example. And this was really interesting because if you look at what's happening with some of these interactions, and so part of it is, you think about a yeast and acetic acid bacteria, these grow together a lot in the environment, especially in fermentation systems. And so what we see is that the importance of the combination is that basically the acetic acid bacteria is utilizing the ethanol produced by the yeast, and it's making a variety of acetic acid and acetate-derived esters. And so Caleb did a really great study of this to analyze the different components and identify what the actual odorant signatures were. And so this is what you can see here. So growing on its own, you get a really high ethanol level. If you grow it in co-culture, you can see the ethanol is depleted over time and utilized by the acetic acid bacteria. So utilizing a mutant to show that this was the attractive element, if it doesn't have the alcohol dehydrogenase, it can't produce these acetic acid or acetate-derived esters. And so ultimately, if you compare a co-culture with the mutant to the wild type, it's more attractive. But by adding back these esters and the acetic acid, what you can see is you ultimately can get to a point where the mutant with these added back components is no more attractive than the wild type. So we're able to restore the attraction of the mutant by adding back these components. But the question is, why? So is there a relevance of this to the host? And initially, the results we had were a little confusing, because if you look what happens in terms of development of larvae, they develop much better on the mutant than they do the wild type. And we think that's because there's somewhat of an inhibition of the acid. So it is somewhat detrimental and does cause them to grow slower. It also impacts yeast populations, so that these are probably getting much higher levels of yeast. And so that could be a effect. But this larval benefit they get isn't worth the consequences of this association, because if we look in sort of a natural rearing environment, we see that there are fewer eggs laid and then also fewer adults emerging. And this is particularly the case, and this is because the levels of ethanol are also impacting the fly. And so if the amounts of ethanol are too high, it's also inhibitory. And this is especially evident if we look what happens in the context of rearing in a more natural environment. So the acid levels might be inhibitory to the yeast, but as they're consuming the ethanol and increasing the acid, it's also creating an environment where opportunistic fungi can't grow and flourish. And so what happens here with the mutant, you don't get any adults emerging, because essentially all the larvae are killed and outcompeted by these opportunistic fungi. So a variety of Aspergillus and species like that overtake the petri dish in the absence of the acid producing. Whereas the acid production here, by preventing the opportunistic fungi growth, allows the larvae to develop, even if they're doing it more slowly than they would with the wild type acetic acid bacteria is present. And so what this highlights, and what I want to stress here, is that how emergent properties of the microbiome can have important influences on behavior and physiology. And so what I've shown you all together is that the interactions between the microbiome, host, and environmental factors can independently impact the fly behavior and physiology, but their interactions are also really important. And we're just at the beginning of sort of understanding what's happening in this context. So I want to thank the people who worked on this project and my group. So Rose has worked on the odor and binding protein. She's also worked with Sandra, who's an undergraduate from the University of Connecticut, who, related to what Dr. Obalaji just talked about, we see that in terms of this melanization response, chemicals like glyphosate can actually inhibit that. And so that's one of the, we've got a small project on that in the lab where we're trying to understand some environmental cues like that. Danny in my lab works on nutrition and microbiome. Madison and Alex, Benji and Taylor, our other graduate students, collaborators, Caleb Fisher, who's now a professor at Georgetown College, and my colleague, Brian Weiss. And with that, I'd love to take some questions, both in the chat now, but then also any other questions you guys may have later on. Thank you.
Excellent. Many thanks, Nichole. Appreciate it. Really, really beautiful work. So, some questions.
Stop so I can see people.
Yes. I can see my beautiful African background, which I'm very proud of. Clearly. So from Miriam Adeniki Saladin and Helen Kwanahi, they're really asking about, you had mentioned studies where you focused on male flies, and as they rightly point out, probably want to look at both. So in your study, influence of microbiome on the resistance, for example, to systemic ECC5, you only use male flies. So have you looked at female flies? Can the results be extrapolated to female fruit flies? And more generally, do you see sex differences?
Yeah, so in terms of the melanization response, it's a weird artifact that for whatever reasons in Drosophila immunity, systemic infections are largely done with males. And we, in the gut, we largely use females. So I would say all fly immunology work, and even currently how that's influenced microbiome, it's something to look at and consider, because the guts of females are larger. And so I think that's why we've done a lot of gut studies are focused on that. But we definitely know that there are sex effects both on gut, but then also on immune responses. So in terms of like how quickly they amplify the immune response. So in terms of the melanization, we know that the same sort of clotting and wounding is the same in females as we see in males. And so that we do see at least, so I would say the kinetics are slightly different, but that we do see at least a similar phenotype. But yeah, that was a really important aspect to consider. And it's something that actually at Hopkins now I have a colleague who's very into aspects of related to this. So he's been pestering me a lot about some collaborations we can do to look at this in more detail.
So I just want to follow up on that. So the fields choose one sex versus the other to, that's surprising.
It's, well, I think it's just, so I would say so I started out in I've always worked in host microbe interactions, even before actually we termed microbiome. And I worked in Lepidoptera, so moths and butterflies, and I was actually really interested in toxins, so Bt toxin and how the microbiome influenced that. But when I finished my PhD, it was obvious that going further into microbiome, I needed to one, know more about immunity, but also two, a model system would be really helpful, because the system I worked in, this is pre-CRISPR days, 30 chromosomes in a Lepidoptera and 1 generation per year in the model I was using. So switching to flies was a really valuable system. The lab that I went to, Bruno Lemaitre, comes from the Jules Hoffman School of fly immunology. And you may know Jules' name from the Nobel Prize a few years ago. And so I would say what you realize is that there is just habits that form within a field, right? And so the protocol of the lab was to do it this way. And so the nice thing about that is you can look at it and say, well, why aren't we looking in females too for this response, or why do we do this? And so I think now you see that with our first handle on what's happening with these responses, is expanding that now to ask these important and more detailed questions. But yeah, that's kind of where it came by. It was just, I would love to go back and ask, like, why did you first start doing it this way? But like for longevity studies, a lot of times people do these in you'll see if you look at them, they can be largely focused on males because they don't lay eggs. So you don't have to worry in your tubes 100 days over a lifespan study what's happening. But now we know, we go back and we correct this and try to look for sort of the nuances now of the system so that we can understand it better.
Right. So Victoria Alaseni wanted to know how you got those amazing images, the melanin images from such tiny flies.
So camera attached to a dissecting microscope is really what it comes down to. And so that's how we do that. Yeah, it's not easy. You can actually see it pretty nicely under the dissecting scope just by your eye, but yeah.
Cool. Romano Bello wants to know how feasible is immunotyping in Drosophila, and they have particular interest in detecting tumor markers in cancer, how you would do this.
So there is a lot of really beautiful work, both at the gut level and then also even with systemic responses to using flies as a cancer model. And so I'm going to deflect to that. I'm happy to help you find some papers and maybe start with some review papers for it. But there really is a lot of really nice work. And for me, what's exciting is now there are some people starting to look at how microbiome impacts on the gut can be part of that. So we can look at hyperplasia. We can look at different, pathways that are important in mammalian responses and try to understand them more, but also looking at, people have started to look at what happens in blood cell responses and things of that nature as well. But I will have to defer you to those studies, but I'm happy to help point you in the right direction.
Yes, there are some fabulous studies on those.
Mm-hmm.
From Adavin Bismarck Dekora. Nice presentation. Actually, there's a lot of that here, so you should make yourself feel good by looking through that. Looking at the immune composition of the fly, yeah, it's a similar one. How do we use it in cancer immunotherapy studies?
Mm-hmm.
So actually, we're going to put those two together. So I'm going to jump to Asayam Wanbo Asaranotor. I apologize if I'm murdering that name. Can Drosophila be used to study the effects of microbial toxins on humans? Microbial toxins.
So I would say, so actually we have a project in my lab where we're trying to understand human probiotics. So can we take something like E. coli Nichole, which was identified because it prevents Shigella toxins. And understand what makes a probiotic a good probiotic. And so that's one of the projects that we're studying. So yes, we can understand toxins that can disrupt gut tissues. I think especially anything that can impact, whether it's, cell junctions in a gut, kill cells through different secretion of factors. I think the fly system is amenable to that. I think the nuances are finding microbes that can colonize the gut to do that. So for example, this E. coli Nichole that I mentioned, one of the reasons we're intrigued about it is that we've tested a lot of different E. coli in the fly gut. And I would say in most cases, they are really pathetic at colonizing a fly gut, which is an interesting question. It's an interesting aspect of its own, given that we know E. coli can be such a great colonizer of different mammalian guts and especially human guts. But for some reason, this E. coli Nichole is a good colonizer. And we're really curious about understanding why that is. And if that tells us anything about both how the fly gut differs from mammalian gut, but then also what are the similarities and what are things that we can use to probe that more. So I think there's a great potential and in some cases untapped ways of understanding that. But absolutely, if you're looking at microbial factors that can disrupt guts and impact, I think, tissues in general, because we're now starting to understand that what happens in the gut, just like with my OBP study, doesn't mean it stays there, right? So the impacts that we see in the gut can extend elsewhere, and whether it's gut-brain axis, we've got some dopamine producing bacteria in the gut or things like that. There's a lot of great things to study for this with this system that we're just beginning to look at. So
Cool.
Go at it. We'd love to see more studies from some of these great ideas.
Yeah, I agree. I'm actually taking notes on some of these ideas. I'm going to be stealing some of these ideas. Last question from Julia Cordero. Great talk. You showed that the immune deficient flies have a wide spread dysbiosis bacterial load in the gut when you're immune deficient, which was very impressive. I have to say, it was very cool the way it spread out there. Is this the action of systemic immunity or gut immunity? Yeah,
probably both. This was looking at a whole body mutant. So yes, it didn't have the systemic immune response. I think if you look at Aidan Foley's work, he's done some really beautiful targeted IMD, both activation and knockdown in the guts. And so I think it's going to be a combination. There's going to be local factors that are really important because there's that one-on-one interface, right, between the gut and the microbiome. But obviously, systemic factors also have an important role. And for example, with things like the negative regulators I talked about, there we know it's both what's happening at the gut, but then what's happening systemically. That's important for dampening that response and keeping the homeostasis of the insect. Keep maintaining homeostasis in general. Mm-hmm. Yeah.
Sure. Cool. All right. I'm going to have to end these questions. Carlos. So thank you, Nichole. Much appreciated.
Thank you. I'm happy to follow up with anything. We're also recruiting postdocs. So if you have interested or know someone, let us know.
Oh, really? Would you take an old guy like this?
Sabbaticals, maybe. More than happy to.
Cool. Thank you.
Thank you.
All right. And check the chat and Slack. Carl.